Hello everyone, welcome to this lecture on cluster differentiation, CDs. Cluster differentiation can feel like a tricky topic, but today I will break it down in a simple and logical way. Let's begin with CD1A. Imagine if your immune system had a special tool just for identifying lipid antigens. That tool is CD1A. Now, where do we find it? On Langerhans cells, the immune cells on the skin. CD1A is a surface glycoprotein found on these cells. And what do? They present lipid and glycolipid antigens to the T cells. At this point, you might be thinking, wait, isn't antigen presentation the job of MHC molecules? You are right. MHC presents the proteins or the peptide antigens. But the CD1A is unique. It specializes in presenting lipid-based antigens to the T cells. Here's a clinical connection. Can you think of one condition where CD1A is crucial for diagnosis? Exactly. The answer is Langerhans cell histiocytosis because it is expressed on the Langerhans cells. Why? Because CD1A marks Langerhans cells, and in this disease, Langerhans cell histiocytosis, there is an uncontrolled clonal proliferation. We see increased CD1A expressions. So, what lipids does CD1A present? It presents the self-lipids to maintain tolerance, reminding the immune system not to attack its own normal cells. And it also presents pathogen-derived lipids like those from the mycobacterial cell wall to activate the immunity. That's why next time you see a CD1A stain in pathology or at exams, the first condition to come to mind should be Langerhans cell histiocytosis. And what else do we see in this condition? The cells are CD1A positive, S100 positive and Langerin positive. And on electron microscopy, they contain Birbeck granules. Birbeck granules. What are they? Those are distinctive rod or tennis racket shaped cytoplasmic granules. So to remember, CD1A is more than just a marker. It is a functional molecule that helps bridge lipid immunity and the immune system and also for clinical diagnosis. Now let's move on to CD3. CD3 is what we call the universal T cell marker. It is an essential part of the T cell receptor complex, and that means you will find it on all mature T cells throughout their life cycle. Why is this important clinically? Because CD3 is the generic T cell marker we use in immunoflow cytometry and immunohistochemistry to identify the T cell populations. Mechanistically, CD3 doesn't work alone. It associates with the T cell receptor and also with co-receptors like CD4 or CD8. Together, they facilitate binding to MHC molecules, whether it is MHC class 1 with CD8 or class 2 with CD4. So what does this mean functionally? CD3 is critical for T cell activation and for initiating the immune response. Without CD3, the T cell receptor cannot effectively transmit signals after recognizing the antigen. So, Whenever you think of T cells, whether CD4 or CD8 cytotoxic cells, remember that they all share one in common, that is CD3. Now let's move on with our next CD. Now let's move on to our next marker, CD4. CD4 is the classic marker of T helper cells. You can think of it like a label or a badge that identifies these helper cells and their different functional teams. This is the T cell receptor and this is the CD4, which helps to identify the T helper cells, and it's like a badge. But which teams are we talking about? The CD4 marks all the major helper subsets. The T helper 1, T helper 2, T helper 17 and T regulatory cells. That is the Treg cells. So what do all these have in common? They are all CD4. They recognize and bind MHC class 2 molecules. This is the MHC class 2. And where are MHC class 2 molecules found? On the professional antigen presenting cell. The red one is the MHC class 2. This blue one is the T cell receptor. This purple one is the CD4. The MHC class 2 is present on the antigen presenting cell. That is this big purple cell. What is the professional antigen binding cell? It is the macrophages. It can be a macrophage. It can be a B cell or a dendritic cell. This interaction is critical because CD4 plus helper T cells are the coordinators of the immune response. They don't directly kill affected cells. Instead, they give the orders. They activate the macrophages. They give the order so that it can stimulate the B cells. So whenever you hear CD4, think of immune systems team leaders working hand in hand with antigen presenting cells like these through MHC class 2. Let's move on to CLL, that is chronic lymphocytic leukemia. To elaborate further and to understand better, let's see what does the word CLL, chronic lymphocytic leukemia, mean. Chronic is something which has been there for a while. Leukemia is a proliferation of cells, neoclonal proliferation, and L, chronic lymphocytic. What does the word L mean? It means that the cancer arises from the cells of lymphoid. That's why it is lymphocytic. Now, lymphoid cells give rise to three main lineages: the T cells, B cells, and the natural killer cells. But in chronic lymphocytic leukemia, the malignant cells actually arise from only the B cell lineage. So here's the question. If it's really a B cell cancer, then why do we use the broader term lymphocytic, which could mean T, B or NK cells? The reason is historical. In the early days, before we had immunohistochemistry, diagnosis relied only on microscopy. Under the microscope, the tumor cells looked just like small mature lymphocytes, that is, with dense nuclei and scanty cytoplasm. But with just morphology, you couldn't tell whether those lymphocytes were B cells, T cells or NK cells. So they simply named it as lymphocytic leukemia. Later, with the advent of immunohistochemistry, researchers discovered that these cells expressed B cell markers, confirming that CLL originates from B cells. So today, even though we still call it as lymphocytic, we know it is really a B-cell neoplasm. So this is in short about CLL, which is a B-cell neoplasm. Now let's turn to CD5. Normally, CD5 is a T-cell marker. It is expressed on both helper T cells as well as cytotoxic T cells, CD4 and CD8. CD5 is rarely found on normal mature B cells. But here's the key. In chronic lymphocytic leukemia or CLL, the malignant B cells aberrantly express CD5. In other words, they inappropriately display a T cell marker. This is not seen in most other conditions, lymphomas. So the presence of a CD5 on a mature B cell becomes a diagnostic clue. So it strongly points towards CLL, chronic lymphocytic leukemia, or SLL, that is small lymphocytic leukemia, which is its tissue counterpart. This is why we often say that CD5 is positive for B cells, are pathognomic for CLL and SLL. So in short, when the CD5, which is supposed to be present on T cells, is aberrantly expressed on the B cells, then it is malignant B cell lymphoma in CLL, chronic lymphocytic leukemia. Here we are going to see the comparison between CLL CD23 with mantle cell lymphoma. We previously saw that chronic lymphocytic leukemia is a B cell malignancy that interestingly expresses CD5, a marker we usually associate with the T cells. But here's another important point. CLL is also CD23 positive. Now contrast that with the mantle cell lymphoma. It is CD5 positive, but it is CD23 negative, unlike CLL. Let's understand why this difference exists. The germinal center is the center of a normal lymph node. The outer zone is a thin ring which is surrounding the germinal center. The naive B cells enter the lymph nodes through the first zone, that is a mantle zone, and then into the germinal center. From there, they undergo proliferation, somatic hypermutation, and differentiation into mature B cells, capable of producing the antibodies. So, to make it even more clear, mantle zone is like a waiting room, whereas germinal center is the place of maturation. What happens in mantle cell lymphoma? In mantle cell lymphoma, a B cell in the mantle zone acquires a genetic abnormality, like a translocation of 1114 happens in mantle cell lymphoma. Translocation leads to cyclin D1 overexpression. This abnormality drives unchecked proliferation right in the mantle zone before the B cell can enter the germinal center. As a result, the cells keep multiplying in the waiting room without maturing. They remain CD5 positive, just like CLL. But because they never pass through the germinal center to fully differentiate, they do not express CD23. So, mantle cell lymphoma has CD25 positive cyclin D1 overexpression but CD23 negative. So you might think like in the waiting room, one restless B cell refuses to wait its turn. Instead, it starts making endless copies of itself, overcrowding the lymph node and never entering the actual germinal center. This is what happens in mantle cell lymphoma. So since it never enters, it is CD5 positive and not CD23 positive. Whereas what happens in chronic lymphocytic leukemia, CLL? On the other hand, this represents a malignancy of mature B cells. These cells have already entered the germinal center, undergone differentiation, and are more advanced in their journey. Because of this maturation, they express CD23. So CLL is CD5 positive as well as CD23 positive. But mantle zone is CD5 positive but CD23 negative because it proliferates early and never matures. So a summary, mantle cell is CD5 positive but CD23 negative and cyclin D positive. CD8, it is a marker of cytotoxic T cells. It is the primary marker of cytotoxic T lymphocytes. These cells are the effector arm of the immune system, responsible for directly destroying target cells. Which cells do they act on? Virtually all nucleated cells in the body express the MHC class 1 molecules. When a normal cell is infected with a virus, transformed by malignancy, or is foreign, these abnormal peptides are presented on MHC class 1. How does CD8 help? CD8 binds to class 1, MHC class 1, stabilizing the interaction between cytotoxic T cell receptor and the antigen. This recognition activates the cytotoxic T lymphocyte to release perforin and granzymes, initiating apoptosis. So diagnosing T cell lymphomas where aberrant CD positivity can also be a defining factor, and it can be used in diagnostic utility. Now let's turn to CD10, which is a key marker associated with ALL, acute lymphoblastic leukemia. First, what is ALL? ALL stands for acute lymphoblastic leukemia. Acute means the disease progresses rapidly. Leukemia means malignant proliferation of the WBCs. Lymphoblastic tells us that the malignant cells are lymphoblasts, immature precursor lymphoid cells. What can a lymphoblast become? Lymphoblasts are precursors for B cells, T cells, and also NK cells. Though for simplicity we can set NK cell aside. So ALL can either be from B cell or it can be from precursor T cells. Which type is more common? B cell ALL, 85% of cases, especially in children. T cell ALL is less common. So till now this is about acute lymphoblastic leukemia. Where does CD10 come from? CD10 is also known as CALLA, common acute lymphoblastic leukemia antigen, a cell surface enzyme which is expressed on precursor B cells in the bone marrow. Therefore, B cell ALL is CD10 positive, but whereas T cell ALL is CD10 negative. Because B cell ALL is so much more common, when we generally say ALL, we often imply B cell ALL with CD10 positivity. T cell ALL is not CD10 positive, but that is not common. Therefore, ALL is positive for CD10. Thank you for listening. If you found this teaching helpful, I would appreciate if you give me a thumbs up. I mentor medical graduates and USMLE aspirants. If you would like my guidance, feel free to reach out in the comments.